Thank you very much. In this lecture, I'm going to talk about genes and flies. And the reason why I'm interested in genes and flies is that flies are very interesting. They have extremely interesting properties in the sense that they're well, I think I'll take my things away. And here we have Superfly. You'll remember Frankenstein wants to construct an angel. And if we could do what we can do with flies, with human beings, then Frankenstein would be terribly excited. And let me show you here. You will remember The Hitchhiker's Guide to the Galaxy. No, he wouldn't be interested in the robot.
I suppose you'll want to see the aliens now. Do you want me to sit in a corner and rust, or just fall apart where I'm standing?
Show them in, please, Marvin. Ford, hi. How are you? Glad you could drop in.
You could see why Frankenstein would be interested. Now, we can do with flies that sort of thing. Now, to help me talk about the flies and to show you the flies, a friend of mine who does research on them in Cambridge has come along, Michael Ashburner, and Michael has brought his flies with him. Now, the first thing we're going to do is Michael's going to show you some normal flies, and we've got a volunteer, Krishna, who's had a little training already because it's a little delicate handling flies. Krishna, would you come and be with Michael, and he can instruct you how to handle these flies.
Hello. Well, what we're going to do is to put some flies to sleep so that we can look at them alive under the microscope. And Krishna's going to help me to do this. To put the flies asleep, we knock them out with carbon dioxide, and I'll just turn on under here a cylinder of CO2. And you can see the gas bubbling through this water here. I put water in simply to keep the air moist so that the flies don't dry up. Now, Krishna, here's the plate with the carbon dioxide coming through, and here's a bottle of flies. Perhaps you can knock those out for us. That's right. And take the top off. Don't worry if you get away. They knock them down. They won't bite you. That's fine. Put the top back on.
Michael, while they're going to sleep, what are they living on?
They're living on a food which has a yeast extract in it. Normally, in the wild, they live on rotting fruit, but in the lab, it's much easier to keep them with the yeast extract. Now we'll put those under the microscope, and Krishna can come and have a look at them. I'll simply quickly find something. We should have that up on the screen. Now you can look. And I can go to the screen and with a pointer show you what a fly looks like. You see this fly has a head, a thorax, and an abdomen. The abdomen is segmented, and Lewis will talk about that later. Has 3 pairs of legs and a pair of wings. And on the head you'll see it has an antenna here. This is a female fly because it has a very large abdomen, which is full of eggs. And I'll show you a male in a minute, and you will notice that the male and female can be told apart because in the male, the back end of the abdomen is very black. Now, Kristen, could you just move the plate, and we'll see if we can find a male. move his wing out. And there, I think you can see the black pigment on the back of the male fly. These flies are still alive, they'll be twitching a bit, and here you can see quite well its antenna.
And the antenna they use for smelling?
Yes, the antennae are used for smelling, particularly so that they can find food and other flies. His wings moving in.
All those hairs on the legs?
These hairs on the legs are also sensory, and I don't know precisely, but they presumably use those to sense air currents and the like.
Okay. Thank you very much indeed for that, Krishna. That's very kind. Just let me show you, first of all, thank you very much. And while Michael, those flies are perfectly all right. They sort of recover very happily from a little whiff of carbon dioxide. Just let me show you on this model some of the things that Michael was pointing out. Of course, the wings are quite clear here. Can you see the antennae at the front here? These are the things that they smell with at the front here. You can also see the legs, the 3 pairs of legs. And I just want you to have a look at this structure here, which is just behind the wind, and that's the gyroscope, which is involved in them flying. And you can see the abdomen there. And I think, and of course, this is the bit that's concerned with feeding. Those are the main normal structures in the fly. What we want to look at now, and what Michael's going to show us, is some mutant flies. And the concept of a mutant, it sounds as if it's something awful, but I want to tell you, in a sense, we're all mutants. Compared to the animals which we evolved from, we are mutants. In other words, mutation, in the sense we're using it here, it just means that there's a change in the genetic information so that structures look different.
Okay, I'll show you 2 sorts of mutant fly. And these were provided for me by Peter Lawrence, a colleague of mine in Cambridge. Let's put them to sleep.
Are these ones that arose spontaneously?
These can arise spontaneously, but in the lab we usually induce them with X-rays or with chemicals. Well, this fly is upside down, doesn't matter. And you can see that instead of, if this fly will stop moving, the plate will stop moving. Let's find another one. Sorry about that. Stick it down. That instead of having 2 antennae on top of the head of this fly has legs. And it's a mutation which changes the antennae into legs, which we call antennapedia. Now, the second mutant I will show you is in some ways even more dramatic.
Of course, one of the virtues of flies and why you can do genetics on it so nicely is they have a very short life cycle, from egg to egg in 9 days. So you can breed flies very, very fast.
These flies are now out. We can have a look at those. And you can see that these flies are quite extraordinary because instead of having, any normal fly, 1 pair of wings, these flies have 2. Not sure that's quite in focus. That's a bit better.
That's amazing.
And you can see that there are 2 pairs of wings on this body, due, again, like the previous fly, to a mutation. These flies cannot fly because the structure which forms the second set of wings is, in fact, that organ which would normally act as a gyroscope for the fly and which keeps it balanced whilst flying.
So it's this structure here, the gyroscope, that is turned into a wing.
That's
right.
Right.
These genes that you're talking about really are major controlling genes because they actually change one structure into another.
That's correct.
They can change an antenna into a leg,
Yes.
and they can change this gyroscope into another wing. And these are single genes. which bring about these major changes in the structure of the fly. Incidentally, this model here, which is, of course, a large one, that's about 100 times larger than the flies that Michael's been looking at under the microscope. The other feature which makes the flies so impressive from the point of view of Frankenstein and all developmental biologists is not only that we know so much about their genetics, but there's something very special about their chromosomes. And Michael's now going to isolate their chromosomes and show it to us under this microscope, but he has to actually do the operations under that dissecting microscope there. Michael, will you explain to us what you're actually doing?
Okay, whilst here I have a bottle of a culture of Drosophila with a lot of larvae in. And what I'm going to do is put one of those larvae in a small dish under this dissecting low-power microscope in a bit of salt solution. Just take them out with a brush. These larvae are feeding on yeast. In fact, there are about 4 in now, I think. And you should be able to see them, I hope. I move this under the dissecting microscope. And these larvae have a definite front end where a mouth is. You can see perhaps that there are black mouth parts at the front end of the animal.
Can't quite see it, Michael.
No, main focus.
That's better.
That's better.
That's the front part or the back
That's the
part?
front. That's the front part with the mouth hooks moving. And at the back, you can possibly see, if I move one of these larvae out of the way, there are 2 orange structures, which are the back of the air pipes through which the larva breathes. So what I want to do now is to dissect out the salivary gland from these larvae and make a preparation whereby we can look at their chromosomes. The reason why we use the salivary gland is simply that it has the largest chromosomes. So I'll just sit down and do that.
Can I just show you what you saw with Michael's larvae there? There's the head at this end here, and then at the other end, the back end, you have those air pipes, which I think you could see on Michael's larvae, and that's where the air comes in. And of course, this larva is very similar to a caterpillar in that sense, isn't it, Michael? That's
Yes,
true,
that's
isn't
quite
it?
right.
Okay. Have you? How are you?
I think I have a dissected larva now.
Okay.
I can get it on the screen. And I'm just going to tease out with my needles, up there somewhere, the salivary glands. And there they should be. Can you see those?
Yes.
A larva trying to eat them. We can move him out the way. They're not particularly carnivorous, but I think they're just curious. So that's a pair of salivary glands. And I'm going to transfer those onto a microscope slide with a pipette. I can find the right pipette. Suck them up in a little fluid and put them on a slide. And in order to see the chromosomes, we have to stain them. And I have a red stain here. So I'll add a bit of stain, like that. And very quickly, that will stain the chromosomes so that we can see them. But first, we have to squash the nuclei and spread the chromosomes, which I do by putting a cover slip on top and simply pressing down on it. Now, these chromosomes are very small, so that we have to take them to a high-powered microscope in order to see them. So I'll move over to the other microscope on the other side.
You make it sound very simple, Michael.
It is quite simple, Lewis.
You think even I may learn to do it?
I think if you came to Cambridge, we could teach you in an afternoon, yes.
I should emphasize that the special feature about the fly's chromosomes, particularly in the salivary glands, is that they're very thick. And that's why we're going to be able to see them so nicely. It's a unique system in this sense. And the reason is that when the cell divides, it doesn't split into two. When it grows, it doesn't split into two. It goes on growing. And the chromosomes, when they double up, the genetic information of the chromosomes and the genes, when they double up, they just lie next to each other. So you get a nice thick chromosome. And so what what you can see here, perhaps you're going to explain that to us.
I'll explain it. Just get that focused. About it. Yes, here we have a squash of the chromosomes of the salivary gland, which I just dissected from the larva. And there are 5 chromosomes, and you can see some of them very clearly on this preparation. Here's the end of one chromosome. It goes round just off the screen, winds its way back to here. Here's the end of a second, the end of a third, the end of a fourth. The end of the fifth is rather tangled up in here.
Michael, I think I should make clear that these are the chromosomes from a single nucleus of a single cell. So this is the genetic information. In principle, if we could open up one of your cells, one nucleus, if they were as thick as if your chromosomes were as thick as this, you'd also see patterns like this.
Yes, that's true. And here you see that these chromosomes have a very characteristic pattern of these darkly staining bands right the way down. And in principle, the genes are contained within these bands, and as a rough approximation, there's one gene to every band.
5,000 bands?
About 5,000 bands, yes.
5,000 genes control the making of the fruit fly.
At a rough approximation.
Okay. Can you tell us, if each one of these is a gene, can you tell us a gene that does something interesting? For example, you had those mutants. Which is the gene? Can you point to us which gene it is?
The gene for the first mutant I showed you, with the antennae with the leg transformed from the antennae, that gene is in this region here. The gene for the four-winged fly is up in this region here. There's a gene down here, for example, which when mutant gives the flies a white eye color rather than red. And perhaps lastly, I can point you out my favorite gene, which is a gene up here, which codes for an enzyme called alcohol dehydrogenase. And the reason why flies have an alcohol dehydrogenase is that they live normally in fermenting fruit, and without this enzyme, they would get drunk.
How wise flies are.
Wise flies are.
Michael, I'm very grateful for your help. Thank you very much indeed.
Thank you.
We now want to look a little bit more. Now we know we have this capacity to do genetics with the flies. We can name the genes. We can actually see the genes on the chromosomes. We can see that there are certain controlling genes that could bring about major changes. We want to understand a little bit more about the development of the fly. And on the same scale, with the fly and the larva, here we have the egg, and these two little structures at the front of the egg, that's the way the egg breathes. And you go from the egg through to a larva, and then the larva metamorphoses into the fly. But first of all, just let's have a look at the early development of the larva. And here's the early development of the larva, and what you can see is that segments are forming. It's forming a series of repeated units, which are really the basic body structure of the larva and of the fly. And I think you can see these repeated units with the grooves between them very clearly there. And I think if we go back to this model here, you can see once again that there are a series of repeating units with little grooves in between them. So the larva is segmented. Now, this larva, we saw, eats, feeds, and then it goes at a particular time, it undergoes metamorphosis into the fly. And we can see this here. It's just like a caterpillar turning into the butterfly. And here it's emerging from the pupa. It's undergone metamorphosis. And that sort of rather strange-looking larva has turned into this rather beautiful fly. And of course, in the case of butterflies, it's even more beautiful. And we need to understand how that happens. How does this larva turn into this fly? It's metamorphosis. It's under the control of a chemical, a hormone called ecdysone. But if you compare its metamorphosis, the change from this to this, with that of a tadpole into a frog, there's a very important difference. Because when the tadpole turns into the frog, large bits of the tadpole, the existing tadpole, actually turn into the frog. Yes, it loses its tail, it grows some legs, but on the whole, it's the tadpole turning into the frog. That's not the case here. Because let me open up this larva and show you what's inside. And what's inside, there are other things inside, but what I particularly want to show you inside is that you see these colored pieces here. They are known as discs. And in fact, the front region of the fly particularly is made from those discs. So let me take out one here. That's the wing disc. There's one on this side of the larva, and there's going to be one on that side of the larva. And this is a sheet of cells, rather dull-looking, not terribly interesting cells, which, under the action of that hormone ecdysone, that will change during metamorphosis into a wing. Let's have a look at another disc. This disc here is a leg disc. And this sheet of cells will change, also under the action of the hormone, into one of the legs. And so what really is happening here is the fly is carrying around inside, so not the fly, the larva is carrying around inside it, as it were, the do-it-yourself parts to make a fly. It's not the larva that's turning into the fly, it's the pieces inside that are actually going to change and somehow, and nobody quite understands how, fit together to form the adult structures. Just to emphasize it again, this is the sheet of cells that's going to form the head and the antenna. So all the parts are in there, waiting to go, waiting to the signal from this chemical, this hormone ecdysone, to change into these adult structures, and in some way, as I say, we don't understand, to join up to make a whole fly. For the abdomen, for these regions there aren't actually discs, but there are these little groups of cells, and those are the ones that are going to give rise to the abdomen. And then the brain, that's the one bit of the fly that actually comes from the larva. Large bits of the larval brain actually end up inside the fly's brain. So it's a curious do-it-yourself kit. Now, when metamorphosis occurs, and when this hormone acts on these discs, it changes the pattern of gene activity. Genes that weren't on before now come on, and they begin to make their messages. And one of the remarkable things about flies is that you can actually see this process. And if we just focus on a couple of genes, so here's the chromosome of the kind that we saw before, and here are the genes, and these are simply known as 87C and 87A because all the genes in the chromosome are numbered and have special numbers and letters. What happens is that during metamorphosis, a gene that looked like that changes so that it looks like that. It puffs. And that puffing is actually an expression of gene action. That shows as a gene is actually being turned on. There's another one here. This one's going, also changing and puffing. These, of course, are really remaining the same. And so you have some genes which were quiet and silent before, beginning to sing their song, give their message. They produce a product which makes another gene turn on. And so you have a whole cascade of genes being turned on or off. And it's those changes that are actually controlling the metamorphosis in the fly. And what I think is particularly nice about that is that one can actually see the genes, the activity of the genes. I've already told you that this one at the front here is the disc for making the antenna. And one of these here, this one here, is the disc for making the leg. But now we've already seen a mutant in which those discs get confused. Because just let's have a look again at that mutant antennapedia. This structure sticking out of the head of the fly is not an antenna. That is a leg structure here. And what happens in the mutant antennapedia is that the antenna is transformed into a leg. And we can actually see that under slightly higher power. You notice that the leg can move. It moves rather weakly. It's a funny place, to have a leg effectively where your nose should be. And here it is again, and actually it's got one of its other legs crossed over that leg growing out of its head. It's not an ideal way for the fly to live, but they survive quite happily like that. And what we want to try and understand is what on earth that might mean. And of course, from Frankenstein's point of view, here we've got a gene, a single gene. Just one gene's being changed. That's all that's happened. One gene has gone wrong, which can change an antenna into a leg. And of course, if one wants to construct new sorts of animals, if one wants to construct angels, this may be a way into understanding how genes control structures. And so the first thing I want to try and understand is, what's really the difference between an antenna, which comes from this disc, and a leg, which comes from this one here? And the way I want to think about it is to go back to thinking about pattern formation. Okay. If I could have 2 volunteers, please. Now, I need quite, let's see who the person in the blue, could you come? And the girl in the yellow jersey there, would you come? Yes, please. Yes? Please. If you just come and stand over here, if you could come and stand over here, I'll show you how we're going to think about this. Just wait for it to come down. So I don't know your names.
Ben.
Ben and Anne. Fine. Slightly tiring task you have to have, and I'm afraid I'm going to ask you to be rather, you've got to be sort of avoid looking at the monitor, you've got to be sort of rather tough, your arms are going to get tired, but you've just got to look straight ahead, otherwise the demonstration doesn't work. Sorry about that, but that's the way it is. Okay, here's the stars and stripes. And if you could come and stand over here, right? No, don't hold it up yet, you'll get tired. I'll show you in a moment. Here's the French flag. You can come and stand next to him, okay? I'll tell you when to hold them up in a moment. Could you hold them up now? Okay, and if you could just have your hand up there so as we can see 2 flags. And I'm going to think of the leg and the antenna as the stars and stripes and the French flag. Let me tell you why. The leg and antenna are really made of the same cell types. And so are the stars and stripes and the French flag, red, white, and blue cells. I next want you to think that the way these patterns have been made is that we've got the same coordinate system here as we have here. In other words, we go along the top row, A1, 2, 3, 4, 5, 5, B1, 2, and so forth. And we've got A1, 2, 3, 4, and along the top here, and then B. In other words, the same coordinate system here and here, except why we get a stars and stripes rather than a French flag is that the cells have different instructions as to what to do in different positions. I want to assume that to be true. And what I want to ask is that true of the antenna and the leg? Do they really use the same positional information, the same coordinate system? And the only difference is how they interpret, how they make use, what the instructions they have as to what to do at different positions. Okay. Because the experiment we can begin to think about then is what will happen if we take a group of cells before they form the flag from the stars and stripes and put them somewhere in the French flag. Let's have a look at that. You've got to be tough now. Look to the front. Don't look at the monitors. Arms getting very tired? Not yet. Won't be too long. There's the French flag. There's the stars and stripes. And the question that we're going to ask is, say we take some cells from the stars and stripes before they've actually made any pattern, and we put them in this corner up here of the French flag. The question is, what do you think they're going to do? Let me ask you. Do you think that they're going to, say I take them from the bottom corner, say I take them from this region here of the stars and stripes, put them up here in the French flag, do you think they'll form stripes? Who thinks they'll form stripes still? Okay. Who thinks they'll form stars? Who thinks that they'll just form that part of the French flag, blue? Okay, just let's understand exactly what we're doing. These cells have instructions what to do in particular positions, so they form the stars and stripes. We now put them up here where they have a new set of positions, okay? They behave according to their position and their genetic constitution, and they form stars. Okay. If we'd put them down there, what would they have formed? Let's see, let's move it. They would form stripes. The point being that if we do this experiment and we have the same coordinate system, the cells remember their instructions but change their positions, then they behave according to their genetic constitution and their new position. Thank you very much indeed for being so patient and holding those up. Thank you very much. Now we do the experiment. What we're going to do now is graft at an early stage some cells, some mutant cells, those of which have the antennapedia set of genes, or that one gene changed. We're going to graft that to the tip of an antenna, just in the same way as we grafted a group of cells from the stars and stripes into the corner of the French flag. Let's have a look at the antenna again. Perhaps I'll just remind you what the antenna looks like again here. Here's the antenna at the front of the fly, these lumps here with this sticking out. And we're showing this in a diagram here. It's a jointed structure. That's the bit joined onto the head. It comes like this. And here you have this bit that it actually smells with at the tip. And what we're going to do now is we're actually going to graft the mutant piece, a mutant tissue, into this region here. And the question is, what do you think it's going to form? Any ideas, do you think, what it's going to form? Any volunteers? Antenna. Okay. Let's actually have a look what it does form. It actually forms leg. And which bit of the leg does it form? It forms the end bit of the leg because there are the claws we can actually see. It's just like the French flag and the stars and stripes. The cells behave according to their position and their genetic constitution. If we'd done the graft, and incidentally the way we do this graft is not by actually grafting, but it's done by using a genetic trick. If we do the graft so that, for example, the mutant cells were here, then it would form leg cell, but it wouldn't form the end bit of the leg. It would form the bit of the leg corresponding to about this region in the embryo. Okay. So what this shows us is, and there are other experiments to confirm this, that all these disks have exactly the same coordinate system, yet they develop into different things. And the question is, why do they develop into different things? Is it because they have different developmental histories? But let me just at this stage say, or ask you, or you may get the impression that this is all very irrelevant to Frankenstein's quest. After all, this is all about flies. And after all, Frankenstein, and probably most of you, are interested in people. And you might argue, well, we don't have discs and we're not segmented, and flies have very little to do with human beings. But just let's find out if that's really true. And I've got a volunteer here who's very kindly come along to help us to actually ask the question as to whether, in fact, we're segmented or not. And here we've got a very nice human skeleton. And I think you can see at once that, at least if we look at the ribs, they seem to be arranged in a segmental manner. Okay? We can see this perhaps better if we turn the skeleton round. And let's just have a look at the back here. Have a look at the vertebrae. You can see the vertebrae of the spine in a beautiful segmental arrangement going all the way back to the very bottom part of your back, bottom part of your spine. And also you can see that they change slightly as they go down. The vertebrae up here are rather different to the vertebrae down here. Also, from in between the vertebrae, you actually get nerves coming out. And those nerves also are, of course, segmental because they come out between the vertebrae. I just want to show you now how those vertebrae develop. And here is a human embryo in which you've got a sheet of cells, and we'll see the segments forming. This is quite early in your own development. And you'll see very nicely here blocks of cells appearing. Here they are. And it's these blocks of cells on either side of the spinal cord that actually give rise to the vertebrae. And as we saw before, here are the vertebrae which those blocks of cells give rise to. They also give rise to other structures, which we'll see in a moment. And as I told you, the nerves come out between them. And I've got another volunteer here, dermatome Dan. And what you've got very nicely marked on here are what the various nerves take messages to and receive messages from. Remember, I said that between the spinal cords, between the vertebrae, there were nerves, and these nerves just go to particular parts of the body. In other words, this nerve here takes information to and receives information from just this part here. This one simply receives information in this narrow segment here. So your body is divided into what are known as dermatomes, which reflect which nerves are taking information to them and receiving information from them. Now, that really is quite important, and it really shows some light on some illnesses. Now, how many of you have had chickenpox? Almost all of you. Were you warned that when you had chickenpox, that you shouldn't go near elderly people? Yes. Some of you were. Now, the reason why that is because chickenpox is caused by a virus. And in children, well, you get a skin irritation and it's not very serious. But in older people, the virus affects the nerve. And the result of the nerve becoming inflamed and affected by this live virus is that you can get a really very nasty and painful rash, which is shown here on this picture here. And you can see that the rash runs in a sort of band like this. There's one there, and it's also running like this here. And that is because the reason it's confined to that region is because if we have a look, if we do a little dance, and I turn him around, and we have a look at his back, it's because that the nerve that's infected is the ones running here, the two that are probably running here and here. And if we just go back and have a look at those there, you've got this strip running along here, which corresponds to the nerves running along here. And it's because just these two nerves are infected by the chickenpox virus that you get the rash localized in this region here. And it's because, in fact, that we are segmented that you have that particular location of the rash. So I hope I've demonstrated to you all that we really are very segmented in many aspects of our body. We would really like to understand how the body actually gets segmented. We don't fully understand that. You saw the formation of those blocks of tissue. What we do know in the fly is that the genes controlling segmentation are different to the controlling genes which make a wing or those which make an antenna rather than a leg. But it's really quite impressive that one can actually get segmentation from a solution. And here are some test tubes with some chemicals in, and which initially are really quite uniform. And they start off quite uniform, but within about 10 minutes, you begin to see bands forming, rather like segments. And after 30 minutes, you can see, for example, in this one, you have extremely nicely segmented structure. Now, I'm not suggesting that whatever these chemical waves or these chemical reactions are is how the insect embryo segments or the way we segment. Nevertheless, it is extremely impressive that starting off with a completely uniform solution in a test tube, you can get segments of that nature. And at least it gives us a suggestion that that may be the way to think about it. So we are segmented, the flies are segmented. What we now want to understand is how is it we've got arms in this part of the body, legs in this part, and heads at the top. Hard to do it in man, but at least we can do it in flies. Because let's go back to thinking about the difference. We've already seen that the difference between the cells, the disk of cells that are going to give rise to the antenna and those that give rise to the leg, the difference between them may in fact simply be due to their life history. Because after all, these are in the anterior part of the larva and these are a bit further back. And you may remember, perhaps the way we can think about this is that maybe there's a chemical concentration difference between the back of the larva and the front of the larva. So that, for example, if the concentration is high there and low there, then what might be happening is that you make a leg where the concentration is that, but at a lower concentration, you make an antenna. And when the antenna gene, something goes wrong with it, you make a leg by mistake. And let me now try and illustrate that to you. So what we're going to look at is a model which is based upon the idea that there's a concentration difference between the back and the front of the embryo. And so what I want you to think of is this is the front end, and that's the back end of the embryo. And if we just slowly reveal, if we look at the chemical concentration, very low at the front, increasing, getting more and more, and high at the back. Okay. Now I want you to think of these controlling genes, these important controlling genes, which are going to be turned on at different concentrations of this chemical. So let's have a look in the first region. No, concentration's too low. No genes turned on. At this concentration, gene 1, then genes 1 and 2 as the concentration goes up, then genes 1, 2, and 3, then genes 1, 2, 3, and 4, 1, 2, 3, 4, 5, and then the last one, in the back end, at the very end, all 6 genes are turned on. And if these control the character of the segments, then we're going to get what is shown here. No genes turned on, we have the front of the fly. As more genes are turned on, other structures appear. And as we go back, we get more and more of the structures. And at the very end of the fly, all 6 genes are turned on. Okay, that's the model. Now let's see whether that fits with some of the observations. Because there's another mutation in which the gradient or this chemical concentration is changed. And let's have a look at that. And let me show you in what way it's changed. Just have a look here. Remember, in the last one, there was virtually no chemical at the front of the animal. This is a very high concentration. Let's follow it through. It's getting less, getting less, getting less, and now it's going up again, going up again. So we've got a high concentration at the back normally, falling to a medium concentration, and then coming up to a high concentration. Hold on. Which genes do you think are going to be on in this region here? All of the genes or none of them? Who says all? Fine. You're absolutely right. Let's go on. Let's pull. All the genes are on. Fewer. 1, 2, 3, 4. 1, 2, 3. And then up again. 1, 2, 3. And 1, 2, 3, 4, 5, 6. Okay, well, I think you can already see what sort of fly this ought to develop into. Are we going to have a head here? Let's have a look slowly. We see some wings. No, we've got the back end of the fly going towards the middle of the fly. And are we going to get a head? Let's see. We get, if this developed, it went all the way through, it would develop into a double-backed fly, as it were. Let's have a look at another example. Let's have a look this time. Okay, that looks pretty normal. Let's go along. Pretty normal, getting more and more. But no, something's changing. It's going down again. So let's see what will happen. You can see now what's going to happen with the genes. Nothing. 1, 1, 2, 1, 2, 3, and then 1, 2, 1, and nothing. What are we going to find here? A normal head? Let's have a look. Slowly. Okay, so there's the head, and let's go to the middle, Bryson. And we'll go all the way through. And there is a 2-headed fly. So you could see that Frankenstein would be very excited, because here we have genes, and remember that most of these are the effect of one single gene, just one of those bands that we saw on the chromosome, which can bring about these extremely dramatic changes in flies. This is an extremely hot topic of research. Now, Nature is the journal most of us try and get our papers published in. I've had many rejected. I've also had some published. But it is probably the most prestigious of all scientific journals. And in the issue of the 17th of December, just a few days ago, there's an article there on exactly this issue of the gradients. It's called A Molecular Gradient in Early Drosophila Embryos and its Role in Specifying the Body Pattern. And you can see in this article that there are various pictures of people trying to understand and looking actually at the gradients in there, exactly in the same way that we've been talking about here. One's made really quite dramatic advances in recent years with respect to understanding how these genes control the form of the fly and also in bringing about these changes. And one of the things that's happened recently is that one's been able to actually isolate these genes. One's actually been able to take the genes out from the animal and actually begin to find out what the message is that they actually contain. And it's really a rather remarkable result. Because if we look particularly now at the controlling genes, at the genes which can turn an antenna into a leg or this gyroscope into a wing, one can begin to find that when one looks at the message, one finds that one part of the message in these different genes is the same. It's as if they sing different tunes, they make different messages, but there's one part which is common. It's rather like them having a common chord, but singing different songs. Now, first of all, that's very surprising. It's very remarkable that you have these different genes, but one part of them is almost identical to a part in the other. So there's something that's common. There's this identical bit in each of them. And what this region is called, this common region is called, is the homeobox. And so you have these different genes in the fly, the controlling ones, each of them having this almost identical sequence. And here we can see that in this gene here, there's a homeobox region, and in this gene here, there's a homeobox region. And you'll remember when we looked earlier, those are the genes which actually bring about those important controlling changes, changing antennae into legs and gyroscopes into wings. So you've got in the fly this conserved region, identical in several different genes, as we saw there, known as the homeobox. Let's have a look at another animal, another insect. And this insect, hold on, I've got a little, is the stick insect. And it's exactly the same basic structure as the fly. It's segmented. I think you can see it's segmented extremely nicely. It's got 6 legs. The male has wings, but the female doesn't, and I better close up because it's quite easy for the male to fly away. And what, of course, you can realize and what you can appreciate, of course, that in addition to the genes which I've been talking about, there are lots of other genes which make this animal much larger and give it its rather characteristic shape. I must also tell you that these identical sequences, the homeobox genes, are not only present in this nice animal, in the stick insect, and in the fly, but they're present in you and me. So you also have, we don't want that to fly away, excuse me. I think I'll just put him in, if you don't mind. Okay, this one can go on crawling. What we have is we have the identical chord, the identical sequence to the ones that we find in the genes of the stick insect, in Drosophila, in birds, in mice, in sea urchins, in snails. Now, when you find a situation like that in every case, in all these different animals, that you find that there's highly conserved cord, this common identical sequence, it means it's something terribly, terribly important. And maybe it will provide an important clue as to why you make arms here and legs down here, and why your head is at the top part of your body. And there are an enormous number of laboratories that are working on that. And of course, that's really a difficult problem. And part of the difficulty arises from the fact that cells are very complicated objects. Embryos and animals are complicated, but cells are very complicated too. And let me tell you why that is. We're beginning to know what the message from the genes containing the homeobox are. And let's pretend we actually know that. And let's assume that that's the product of a homeobox gene. So that we've got the message, and it makes a protein, and there's the protein. But you know, in your cells, there are something like 20,000 proteins. So that protein may interact with another protein, which may turn on another gene, which may make another protein, which may influence the cell membrane, which may change the permeability, what the membrane lets in, which may change another gene, turn on another gene, and so one goes on. One has a whole sequence of events. There's this cascade of events. And so you start off with the product, or what the message from the gene, and it just gives a whole sequence of events. Now, it may be possible to find out what those are, but it may be quite difficult. And to try and give you some sense, particularly in cells where there may be as many as 20,000 different proteins, let's have a look at a sequence which has been made in Japan, in which gives you some sort of feel as to what sort of problems and what sort of cross-connections there might be amongst the interactions of the molecules within the cell. I hope cells aren't as complicated as that, because if they are, it's going to be very hard to find out the link between turning on a gene and ending up with the 5 fingers of the hand or the wing of the fly. We hope it's going to turn out to be much simpler, but there's no way of knowing. And we'll explore this problem a little further tomorrow when we look at the development of the nervous system. Thank you very much.